These are the 12 hallmarks of aging. Aging has really taken off as an area of study in the last decade or so. And one of the things that led to aging being able to actually get funding for studying was aging was actually classified as a disease relatively recently, which makes the availability for funding to study it more. This is one of the highest interest areas of research in medicine right now, is looking at longevity and how we can increase human longevity. The 12 hallmarks of aging, which could be synonymized to the 12 hallmarks of chronic sympathetic autonomic hyperactivity. Number 1, genomic instability. Number 2, telomere attrition. Number 3, epigenetic alterations. Number 4, loss of proteostasis, or the ability to produce and maintain homeostasis of proteins. Mitochondrial dysfunction, cellular senescence, altered intracellular communication, stem cell exhaustion, dysregulated nutrient sensing, disabled macroautophagy, or the disability of the cell to clean itself up, like we just talked about, chronic inflammation, which we talked about before, and dysbiosis. Of these 12, genomic instability is really the base underlying problem. If our epigenome is malfunctioning or not in the correct state, our DNA cannot be read into RNA, and that can't be read into proteins properly. So basically our body is not going to produce the stuff that it needs to function well, and it's going to produce stuff that leads to more dysfunction. This is really cool, you guys. This stuff drives me nuts because this should be all widespread knowledge in the medical community and in the general population, but it also gives me a lot of hope because we have the ability to treat people so much better than we are right now. We have the ability to make people so much healthier than we are right now. All we need is the information and knowledge and understanding to put it into practice. This is what I'm trying to do with these classes, is give people a condensed version of all the important information that you need to treat your patients better and to treat yourself better. There's a lot of confusing stuff in a lot of these research papers. Again, I've tried to put it into the most important understandable English that people can comprehend and put into practice. Vitality genes. This will be discussed in the peptide therapy class. This will be discussed in the genome and epigenome class. It'll be discussed more in the nutrition and supplementation class. But I just wanted to give you guys a quick preview. There's been a huge amount of study looking at longevity genes. 4 of the most referenced health improving longevity genes and their proteins in literature are sirtuin genes and proteins, the mTOR gene and proteins, AMPK gene and protein, and telomerase is a protein, and then the genes that make telomerase. So telomeres and telomerase. The telomeres are the ends of the chromosomal ends. We'll get to that on the next slide here. If you guys are interested in a doctor that's very knowledgeable and that's using peptide therapy in his practice, Dr. Edwin Lee, he's a great guy. I've used some of his slides and pictures here. It's called the clinicalpeptidesociety.com. Great website. So, as sirtuin genes improve health and longevity, mTOR improves health and longevity if it's in the proper concentration and function. Everything in our body is paradoxical, you guys. If we have too much or too little of something, it's typically not good. There's a very finite range of function where things are functioning properly. If it's outside of that range on the upper level or below that range on the lower level, we're going to have problems. This is just a very brief introduction on the epigenome. So what is the epigenome and what does it do? Humans have trillions of cells in our body. All of our cells, as far as we know, have the same genetic material inside the cell. And this is the crazy cool thing about the human body. It's just so incredibly complicated. In each cell, we have about 6 feet of DNA that's wrapped around something called a nucleosome. And then for the genes to be read, the DNA that's wrapped, just think about like a bobbin and thread wrapped around it. If we need certain genes and proteins to be expressed and produced in our body, our nucleosomes and our histones unravel that DNA so the cell can access a little piece of the DNA to read, and then it gets rewound up, and then it can get unwound and a different section can get read. It's incredibly complicated. It's so cool. All of our cells have the same genetic material in them, but different cells produce different things and they turn into different cells. Our pluripotent stem cells are endoderm, ectoderm, and mesoderm, the three types of primary cells that we have when we're first conceived inside our mom's belly when the sperm meets the egg. Those are our pluripotent stem cells, and those can differentiate into other types of cells. Liver cells and kidney cells have exactly the same genetic material inside of them. It's just different parts of that genetic material are being expressed, which makes them into different types of cells with different functions. It's so incredibly amazing. This was first thought about / discovered like in the 1800s when people like Mendel and Darwin and all those people started looking at inheritance. There used to be a big issue called the missing heritability problem, where we used to think that whatever the DNA said is how you were going to be or what proteins are going to produce. And we know that to be patently false now. Only about 20% of that is true. Only about 20% of what the DNA is guarantees that you're going to have an outcome. The other 80% is modifiable based on the cellular environment of our cells, which is based off of what you're putting into your body, if you're exercising, and all that other stuff, your mental health status. The healthier we are and the better things we put into our body, the more accurately our DNA gets read into RNA and then into protein, the less cleanup our cells have to do, the less energy it requires, and the more energy our cells and our brain has to function normally. Chronic inflammation is a really good example of this. Again, one of the hallmarks of aging. Chronic inflammation is not a normal state for human beings, although the vast majority of the country is living with chronic inflammation. People with chronic inflammation, and this is talked about in dry needling literature, manipulation literature, nutrition and supplementation literature, there's a handful of primary pro-inflammatory proteins, cytokines, other stuff that cause excessive inflammation. So interleukin 1, interleukin 6, tumor necrosis factor alpha, C-reactive protein. These are all commonly elevated proteins that cause inflammation in the body that we see in mental health disorders, in physical health disorders. Basically, if you're a regular human being and you're not eating well and you're not exercising and you're not taking care of yourself and you're actually living life, you have stress, you have environmental toxins, you have other factors that are stressing your body and stressing your brain. This tends to lead to a chronic overexpression of the interleukin genes that cause the pro-inflammatory interleukin proteins, the cytokines, interleukin 1, interleukin 6. Those are the pro-inflammatory interleukins. There's also anti-inflammatory interleukins like interleukin 10 and interleukin 17 and some of the other ones. By eating bad food and by not being in good physical condition, we are causing chronic inflammation in our body by expressing genes too often that should not be expressed. They're just being expressed because the cells are in a state of stress and dyshomeostasis or non-homeostatic condition. This is modifiable, and this is why the epigenome is so cool. The epigenome, this is a good picture here. You guys can read the little left, that's just the blurb from the picture, but this is a good visual representation of the epigenome. We have chromosomes inside of our cells. The ends of the chromosomes, or the telomeres, are like the little caps on the ends of the chromosomes. Telomeres, in general, the longer our telomeres are, the more replications a cell can have, the longer the cell life, and the more ability it has to improve health. The longer our telomeres, the better health span and lifespan of our cells. Almost all cells, most cells, have something called a Hayflick limit, or the maximum number of times a cell can replicate. Once the telomere gets gobbled up, the cell can't replicate anymore, it loses energy, and it goes into autophagy where our body gobbles it up. However, if we're eating bad stuff all the time and not exercising and not doing the proper things for us, the epigenome gets modified, which modifies the way that our DNA gets read. So the epigenome controls how our DNA gets read. So again, there's things called histones and nucleosomes. We'll get more into this in the epigenome class. but based off of what we're putting into our body and how we're acting, we get little tags put on. If you think about the double helix, just think about like little marbles getting put, like getting glued onto the outside of the double helix. Those are epigenetic markers. Depending on the little marbles that get glued to the outside of the double helix will determine how the DNA gets unraveled and read and how it gets expressed, or if it gets expressed or not. This is why it's so cool, and this is why nutrition and supplementation work, you guys. It's because they modify the epigenome, which modifies the way that our DNA gets read and how accurate our DNA gets translated into RNA and protein. And it certainly has an effect on the 90 to 95% of our DNA that we have absolutely no idea what it is, the so-called junk DNA, which is obviously not junk. If you take it out, you die. That's not junk. We just don't know how it works. In general, that's how the epigenome works. We'll get into this in detail in the epigenetics and genome class. Here's another picture, same type of picture. So there's the chromosome. You get chromatin that comes off the chromosome. There's methyl groups and acetyl groups and all this stuff. This is a little bit better picture of nucleosomes and histones. The little histones are the little circles, that whole thing. When you get 8 histones together, it can make a nucleosome. That's what our DNA gets wrapped around, the nucleosomes and the histones, and then it unravels to read certain things and re-ravels. It's very, very amazing process. It's incredible that all this stuff happens. But again, what's really cool about the epigenome is it's modifiable. So this is why it's so important to get good epigenetic tests and blood testing done and genomic tested, so we can see, do we have as an individual some type of genetic anomaly that's going to affect the way that our DNA gets read? And if we do have that anomaly, there are plenty of ways to go about making sure or trying to ensure that certain things or certain genes don't get expressed or overexpressed. A great example of this is folic acid. So we mentioned this a little bit before. Again, this is covered in depth in our nutrition and supplementation class and the epigenome class. So just real briefly, methylation is an epigenetic process. You put methyl groups onto the DNA, which changes the structure of certain molecules and things like that. If we want to have a properly functioning epigenome as far as methylation goes, and there's a bunch of other processes, we must have sufficient levels of a couple of things in our body. 5-MTHF, 5-methyl tetrahydrofolate, is the bioactive form of folate. Folic acid is not usable in the human body. You must have a certain type of gene in your body to be able to methylate or alter or convert folic acid into the usable form, which is 5-MTHF, 5-methyl tetrahydrofolate. 30 to 60% of the United States has a gene anomaly that makes it basically impossible for our body to efficiently convert folic acid into 5-MTHF. It's called the MTHFR gene mutation, or methyl tetrahydrofolate reductase gene mutation. It's an enzymatic mutation that does not allow people to convert folic acid into folate. It's really incredible that humans can convert folic acid at all, because it's an alien substance not found anywhere on the planet. And there's only a couple countries in the world that allow folic acid to be put into the food, because most people have taken it under consideration that over 50% of the population guaranteed is going to have a problem with eating folic acid. And folic acid is put in every single non-organic grain that we have in the United States. This is testable. You can find out if you have one of these gene mutations. I do for sure. Just real quick example for me. If I eat stuff with folic acid in it, the back of my head and my ears get itchy within like an hour, and they'll start to flake, and it's all nasty. If I don't eat folic acid, that doesn't happen. The reason is because I do have one of the MTHFR gene mutations, and my body can't efficiently or can't at all break down folic acid into methylfolate, which is the usable form. It's just like putting water, it's like putting a few drops of water in your gas tank every time you fill up your gas. The car's made to run on gas. It's not made to run on folic acid, or not made to run on water. Our body is made to run on 5-MTHF, 5-methyltetrahydrofolate. It's not made to run on folic acid, so it requires extra energy of our body. It requires extra energy of every human's body to be eating folic acid, because it has to be converted through multiple steps into 5-methyltetrahydrofolate. If you're just eating methyltetrahydrofolate itself, 5-MTHF itself, your body does not have to break it down into anything. You're putting the bioactive usable form into your body already. This is one of the things that will lead to elevated homocysteine levels in your body, amino acid dysfunction, cardiovascular disease, mental health disorders, all types of stuff. This is something that 30 to 60% of the United States suffers with, and I guarantee you 0% of patients in the United States have ever been told this by their doctor, because I've never heard this from any doctor. It's very, very disturbing, except for people like Mark Hyman and Dr. Peter Tia and stuff like that. Regular primary care doctors or mainstream medicine don't know this stuff exists, which is absolutely unacceptable. Okay, so to have a properly functioning conversion system from folic acid to 5-MTHF, or for a bunch of other things that need to be methylated, we need to have proper amounts of active folate, active vitamin B12, active vitamin B6, active vitamin B2, active magnesium, betaine, and vitamin D. 70% of the United States population is deficient in magnesium. 95% of the population is deficient in vitamin D. So just based off of those two facts, if you are eating folic acid and you actually do have the ability to break down folic acid into 5-methyltetrahydrofolate, there's a 95% chance that your body can't do it properly because you're deficient in vitamin D. Being deficient in vitamin D does not allow normal methylation of proteins and anything like that in our body, normal methylation of RNA, normal methylation of DNA, if you're vitamin D deficient, which 95% of the United States population is. Our methylation process is not functioning well, meaning we're getting way too many misfolded proteins that aren't doing what they're supposed to do. Our body has to gobble it up and clean it all up. That all requires energy. It drains our autonomic nervous system reserve well of ATP energy. It drains that, and at a certain point, the well runs dry, and then our body has to make really hard decisions on where to use the available energy, because there's not enough energy available for everything. That's a very, very simple, quick rundown of one of the processes in the epigenome that is controlled by the amount of proper nutrients that we do or do not have in our body. This is why eating well makes you healthier, and it's why eating poorly makes you sicker. If you can get your patients to understand the why behind it, they're much, much more likely to follow instructions and do good things for their own health. If they don't, at least for me, if I don't understand why somebody's telling me something, I just basically don't listen to it. I like to know why, and I think this is just a regular human condition. This is what happens, you guys, to 30 to 60% of the United States population. 30 to 60% of the United States population has an MTHFR gene mutation that does not allow them to properly methylate folic acid into methyltetrahydrofolate, and it's called unmetabolized folic acid syndrome. This is something that happens, like I mentioned before, with gluten protein and the lactose protein. If you're eating a ton of folic acid and your body can't break it down, you actually, and it's a little counterintuitive to think about, you actually end up with excess 5-methyltetrahydrofolate levels in your body. I know I just said that that's the bioactive form and folic acid is the non-bioactive form. But what happens, like so many other synthetic molecules that we're not used to eating, they have a higher affinity for the receptors on the postsynaptic cleft side than the natural substance does. If you're eating a lot of folic acid and you have one of the gene mutations, you actually do have methyltetrahydrofolate in your body, but our cells have a higher affinity for folic acids. Then you end up with all of our postsynaptic clefts being being utilized by folic acid, which is not an actual usable form. They're just taking up space and energy. And then we get excesses of methyl tetrahydrofolate and homocysteine and some other proteins and peptides, which just cause widespread dysfunction in our body. It leads to decreased ATP production, hypoxia, mental health disorders, all sorts of stuff. This is fixable stuff for everybody in the United States if you have the proper knowledge and understanding of what you need to get tested for and how you can fix it. And again, this is not information that anybody has. I mean, more or less, 0 doctors in the United States know anything about any of this stuff. There's some of them, obviously, but there's very, very, very few. And basically 0 of the general public knows about this. This is why it's so exciting for me, you guys, is that we have the ability to improve our patients' health and to improve our own health so much more than people think we do. It's just basically not knowing what you don't know, or knowing what you don't know. That's the problem.